The brain can be stimulated electrically to gain information about the working of its various neural systems. When the brain is stimulated, there is a change in the resting membrane potential and the next action potential is initiated. Okay, this causes behavioral changes depending upon which part of the area has been stimulated. For stimulating the brain electrically, electrodes are used. Microelectrodes are made of fine metal wires through which the nerves are electrically stimulated. Macroelectrodes are made of steel wires and are placed either on top of the scalp or inside the brain to record the functioning of the tracts, which are a large number of neurons together. The electrical recordings can be made over a period of time, such as when the animal is recovering from any surgery. This is known as chronic recording. So, what is chronic recording? Which can be made over a long period of time, such as when the animal is recovering from any surgery. Okay, and what are the micro and macro electrodes are made up of? So micro are made up of fine metal wires and the macro are made up of steel wires. Okay, it is either placed on the top of the scalp or inside the brain to record the functioning of the tracts. Okay, if the scientist is focusing to study a particular nerve pathway where the information is going, then the acute recording is done. So for studying a particular nerve pathway, we do a acute recording. These recordings of the electrical signals are then displayed through an oscilloscope or ink writing oscilloscope. So oscilloscope, it helps to display the electrical recordings per unit time. It has a cathode ray tube on which the display is done. It is used to record the activity of one single neuron. When the action is detected, it is amplified and easily heard by the researcher. Okay, so it is used to record the activity of the one single neuron oscilloscope. The next we have ink writing oscilloscope. This is also known as polygraph. The evoked potentials are recorded with the help of the various electrodes that are attached to the scalp. These are needles, pen tips that record the neural activity on a rolling paper with ink. Okay, when the signal is detected, it is amplified and the pens move up and down, writing the action in ink on a continuously rolling paper. Okay, these recordings of the brain are known as EEG. What is this? Electroencephalogram. Ink writing oscilloscope produces electroencephalogram. This helps to detect any tumor in the brain, epilepsy, or study sleep and arousal pattern. Modern techniques use computer to record the information and display it on the screen. Computer helps to display, store and analyze the information that is collected by recording the electrical signal. Then we have chemical stimulation. Okay, there are various ways to chemically stimulate the brain and make legends. Drugs can be one of them, and it is given by injections or by inserting a tube into the stomach directly or into a vein or fatty tissue in body. The chemical activity of the brain can be assessed by using various techniques. One is by using 2-deoxyglucose technique and the 2,2-DG. It is a radioactive drug actually, and when this drug is injected in the animal, the researcher waits till it is absorbed by the brain. Thereafter, the animal is killed and the brain is removed and slices are made. This comes under chemical stimulation. Thereafter, the animal is killed and the brain slices are made. Then we have autoradiography is conducted. That is, the slices are coated with the photographic emulsion. These are then developed as a film. Neurons that have taken the drug appear like black spots on the slide. So they are basically stained. Another useful technique is cerebral dialysis. A tube is inserted into the animal's brain. The animal is then engaged in an activity, and the neurochemicals released while the animal is active are collected and analyzed using the chromatograph. In vivo, voltammetry is another technique that is used to record the neurochemicals released in the animals when they are actively involved in the some activity. Immunocytochemistry is a process which helps to locate the presence of neuroproteins in the brain, and each neurotransmitter responds to its specific receptors during a synapse. And the microionophoresis. This spells microionophoresis is a process that involves ejection of charged molecules used to apply drugs or chemicals to nerve cells. It helps to locate the specific receptors to which the neurotransmitters respond. Okay, microion tophoresis. It spells like macro, then ion, then tophoresis. The next we have stereotaxic lesion. So, when ablation is not possible for tiny structures located below the surface of the brain, researchers use stereotaxic techniques to damage a structure in the interior of the brain. First and foremost, it is important to identify which part of the brain needs to be investigated. The stereotaxic atlas helps to locate the exact point of the brain where the surgery has to be done. And once this is done, the animal's head is placed in the head holder of the stereotaxic apparatus. Once the head is firmly held in the apparatus, the electrode holder is used to insert the electrode cannula into the brain. The electrode can be implanted permanently, and when the animal comes out of the effect of the anesthesia, behavior is monitored. When the study is over, the animal is given anesthesia again. The electrode is removed and the animal recovers from it. Okay, so these were the electrical stimulation methods and the chemical stimulation methods. Now we have neuroimaging. Brain imaging is known as neuroimaging. Structural imaging methods are used to image the structure of the brain. These procedures helps to diagnose any injury or disease in the brain as tumors. Functioning imaging helps to understand the function of the brain. It also helps in diagnosing diseases related to metabolism or brain disorder or disease. X-ray technique. Initially, this technique was used widely to explore the reason behind any neurological deficits. The conventional X-ray did not help much in the observing neural activity of a patient suffering from stroke or neurological disorders. It only provided information about the structures that absorb the X-ray. The cerebral ventricular system and the circulatory system within the brain is examined by using the contrast X-ray. In this technique, a substance is injected into one compartment of the body that absorb X-rays either less than or more than the surrounding tissue. This creates a contrast during X-ray photography. Pneumoencephalography and cerebral angiography are two ways in which contrast X-ray is used. The former is a very old radiographic technique to examine the brain. Cerebral angiograms are most useful for localizing vascular damage and help to detect any tumors. Cerebral angiograms. The next we have CT scan, which is computerized tomography. This is an X-ray technique used along with the computer that helps to view the structures of the body and the brain. There is a huge cylindrical ring in which the person lies down. The X-ray is passed from one side and the X-ray detector is placed on the other side of the patient's head. Slowly, the X-ray and the detector are rotated and the photographs are taken from all angles. All the photos are then collected by the computer and a CT scan of the brain is produced. A number of scans are taken and then they are combined to get a 3D picture of the brain. And all of the photos are then collected and a CT scan of the brain is produced. Okay, neurologists often use CT scans to diagnose blood clots, tumors, sclerosis, or any internal injury, and if surgery is to be carried out or not. Then we have magnetic resonance imaging, which is MRI. This technique is used when detailed picture of the patient brain is to be investigated. This technique uses a strong magnetic field to gather images of the brain in frontal and horizontal planes. Unlike CT scan, it provides more detailed and clearer picture of the brain. This technique helps to know the blood oxygen flow to the different areas of the brain that are active. Okay, the resolutions of the images are high and changes are recorded as they occur in real time. It is safe as no injection is given to the patient. Positron emission tomography PET. PET scan helps to get or detect the changes in the metabolic activity taking place in the brain. It does not provide any structural information about the brain, rather, it provides images of the brain activity, that is, functional brain images. It is an invasive method where an injection of the radioactive 2-deoxyglucose is given to the patient. When it is absorbed by the patient is given a task to do, and while doing the task, recordings are made. The images recorded show which part of the brain was actively involved in the task. PET scans use radioactive chemicals, which is a short shelf life made in cyclotron, a device to accelerate charged atomic or subatomic particles. It is an expensive technique, hence it is mostly available in research hospitals. Okay. PET is expensive and it does not provide any structural information about the brain, rather than it provides the images of the brain activity, that is the functional brain images. Next we have fMRI, which is a functional MRI. It is a less expensive and less risky technique than PET scan. fMRI primarily measures changes in blood flow, which is hemodynamic response. Currently, it is the most popular method to be used in cognitive neuroscience as is for the medical diagnosis. Then we have diffusion tensor imaging, which is DTI. It is a method that identifies those pathways along which water diffuses in the tracks bundles of the axons. Diffusion tensor imaging gives image of these major tracks. DTI determines the location and also the orientation of neural axons in the brain. This method gives an idea of how the different areas of the brain are connected to each other. This is a particular advantage of this technique as compared to other techniques. Transcranial magnetic stimulation (TMS). It is a technique that turns off a part of cortex by creating a magnetic field. It temporarily inactivates the neurons below the magnet. Researchers use TMS as a mapping technique. For instance, when TMS coil is placed on motor cortex and stimulating different regions along this cortical strip, muscle movements and twitches will be observed on the contralateral side of the body. From this, it can be observed how different body parts map onto different parts of the cortex, and also how much cortex is devoted to any particular body part. The main disadvantages of TMS is that it's not very effective in reaching the subcortical structures. Okay, so TMS is a technique which turns off part of cortex basically by creating a magnetic field. It temporarily inactivates the neurons below the magnet. Researchers use TMS as a mapping technique. For instance, when TMS coil is placed on motor cortex and stimulating different regions along this cortical strip, muscle movements and twitches will be observed on the contralateral side of the body. From this, it can be observed how different body parts map onto different parts of the cortex, and also how much cortex is devoted to any particular body part. Okay.